Hello everybody, today we're going to talk about variables. This is one of the most common questions you will see in GCSE Biology paper 6. Okay, so in science experiments, we always want to test how one thing affects another one. And we also want to make sure that our experiments are fair and the results are valid. To do that, we focus on three types of variables. First, the independent variable. This is the one factor you change on purpose. There should be only one independent variable in your experiment. Second, the dependent variable. This is the one thing you measure. Again, you only measure one thing, so your results are clear and reliable. Finally, the control variables. These are all the other factors you keep the same. That way, the test stays fair and only your independent variable, what you change, is affecting the outcome, your dependent variable. So you remember, you change one thing, you measure one thing, and you keep everything else the same. That's how you set up a fair test with variables in your experiment. Now let's have a look at the real example from past paper. The experiment that I'm showing you is about a student investigating how carbon dioxide concentration affects the rate of photosynthesis. We need to identify the three variables we talked about, both dependent and independent, and control variable. In order to do that, we need to read the prompt. Specifically, the first two sentences, which will give you an idea or the clue of what the dependent and independent variables are. So let's get started. A student used an aquatic plant to investigate the effect of carbon dioxide concentration on the rate of photosynthesis. So remember, whatever the student changes is the independent variable. In this case, it's the carbon dioxide concentration. And what does the student measure? The rate of photosynthesis, right? So this is our dependent variable. However, here is an important tip. Please do not just write the rate of photosynthesis as your answer. This is a common mistake students make. You always need to specify how it is measured. So the second sentence gives you an idea of what to be written as your final answer. The production of oxygen gas can be used as an estimate of the rate of photosynthesis. So your dependent variable should be written as number of bubbles produced or number of oxygen bubbles produced. Now, if your exam provides a table, like here, table 2.1, use it. Tables often list independent variables on the left. Here it is the carbon dioxide concentration, and dependent variables on the right. Number of bubbles produced in 5 minutes. Simply copying this correctly into your answer sheet is all you need. Let's talk about the control variable. And keep in mind that the control variables are the factors that are kept the same during the investigation, during the experiment, to make it fair. If we go back to the text, we are told that the student counted the number of bubbles produced in 5 minutes. And that he repeated the experiment with 5 different concentrations of carbon dioxide. So it means that the time for each trial must be the same. So time, 5 minutes, is our first control variable. If you look at the figure 2.2, we can identify at least 2 more control variables. Here we have a lamp, and we know that the lamp is the source of light. Light is an important factor that affects the rate of photosynthesis. The more light you give to the plant, the more photosynthesis happens, and the more bubbles will it produce. Well, during these 5 trials, obviously, the light intensity should be kept the same to make the experiment fair. So light intensity, not the lamp, but the light intensity as your answer should be as one of the control variables. Now have a look at the test tube. Here we have an aquatic plant, and we know that different kinds of plants produce different numbers of bubbles of oxygen because they have different rates of photosynthesis. So in order for this experiment to be fair, in each of these trials, student needs to use the same species or kind of plant. Another thing that he needs to keep in mind is the size of the plant. We know that the larger the plant is, the more bubbles it will produce because the rate of photosynthesis will be bigger. And vice versa, the smaller the plant is, the smaller the number of bubbles will be. So size as well as the type of the plant should be kept the same.